present are more surviving. So if you look at figure 5 here, you can see that this is preliminary data showing that at lower concentrations, the bacteria biofilm integrity is going up and certainly drops around 4 micrograms per milliliters, and then goes back up and starts stabilizing throughout the concentrations. And so this just gave us the information of whether the biofilms themselves are surviving or not. We didn't know what was the composition of the biofilms, if they still had bacteria, or if which bacteria was surviving where. So figure 6 shows that at lower concentrations and in the control, Serratia dominates, and this is the control right here, and this is 8 micrograms per milliliters. You can see that Serratia dominates due to its red coloring. And in the higher concentration, which is 128 micrograms per milliliter, you can see E. coli dominates due to the white colonies that are present there. Using this, we know that kanamycin, while not completely degrading the biofilms, it is affecting the composition of the biofilm itself. From there, now we have like future directions we have to go to, and we want to see if bacterial concentration would, or the antibiotics effects would change if we have a different bacteria added, or if we add a different stressor altogether, if the biofilm concentrations would change or the planktonic concentrations would change. One stressor we were thinking that can be added is biophages, or another antibiotic, or we were just thinking of adding a newer organism to the samples with E. coli and Serratia. So making, rather than 2 species, making it 3.
So one thing I want to tell you guys who are here listening, just to give you a little perspective, biofilms are the more common mode of growth for bacteria. So bacteria like to be in communities. And if you think about yourself, it's much better to be with other people who would be able to help you if there was something going on, both emotionally, physically, everything. So same with bacteria, they prefer to be in communities, and they're single-celled organisms. So, but the way that we've always taught microbiology is that they're independent organisms living by themselves, don't have anything to do with each other. So that's called a planktonic mode of growth. And in order to get that, you have to prevent them from like settling on a surface. So you got to keep them in motion all the time. So that's why, Una said maybe in like like a stream that's running really fast, they would have a hard time settling on something. So they would continue to be in movement. And in order to do our planktonic assays, we do have to shake our cells so that they don't settle down and attach to a substrate. But the majority of the time, organisms are in biofilm. So clinically speaking, that's a little bit more important. And what they're seeing more now is that the infections tend to be more than one organism a lot of times, especially like after people have gotten burn wounds, or you have a hip replacement or something, and bacteria start to grow. They tend to grow in biofilms. And if it's a multiple infection, multiple organism infection, it's much harder to kill. And the idea is, are they helping with one another if there's an antibiotic around? So that's the long-term goal. So we started, Kara, you asked a question about the longitudinal study. So what we did was that we looked at each organism separately first, and then we started looking at them together. So it's not that we're looking at them for multiple days, but just that, we've been at it for a while with these different organisms. And I think the most interesting part in all this work that Sabrina and Eunice have done is the planktonic story. So normally planktonic is easy to kill, and you can see the numbers are like 2 and 4 in that table, and those are kind of standardized numbers across the country. People know that it doesn't take a lot of antibiotic to kill them. But when you get to the biofilms, they tend to take a little bit more antibiotic to kill. So we were really surprised that when we mixed them together, that it took so much antibiotic to actually kill them, not just to suppress them, but to kill them. There's no way we could give a human being that kind of antibiotic without major side effects. So typically people get antibiotics at the very low concentrations. So it's kind of unreal. But the thing is, you don't have to give so much antibiotic so that the bacteria die within a person, because a person, remember, has their own immune system. So if you can prevent their growth, the immune system can do quite a bit itself. So that's kind of a very interesting thing that we've discovered. The biofilm story is not that unusual. So, they tend to become very tolerant to things that are insulting them, like antibiotics, ultraviolet radiation. And, I think they just become very used to it. And then they don't really die off. So you can see the curve just kind of tends to plateau. Sometimes it'll rise, sometimes it'll come down, but it can go on and on and on like that. So that's kind of where we are right now. We'd like to, I think it's a good point, Dr. Winkler, that maybe we can try putting the antibiotic and leaving it there for more than 1 day. So, they grow the cells, add the antibiotic, wait for a day, and then assay. Maybe if we waited for more days, because typically you take an antibiotic for like a week or something, maybe we would see some different results. So sorry to give you for the interruption, but I just wanted to get you a little bit of a perspective.
Thank you so much, Dr. Gulati, for your input.
And if any of you students are interested in this research, do come and see me.
Yes, it is definitely a research I would really recommend. It's really interesting, really fun, and plenty of room for learning and growing and your new techniques and lab skills.
Also, if you do want to just see what we do, we are fine with showing you guys around if you are in the middle of the experiment. So we are on the 2nd floor of the science building. If you guys want to just come and see, we'll just get a signature saying that you guys signed for safety.
Well, enjoy the rest of the conference, guys. Bye.
Thank you, Dr. Gulati.
Thank you so much.
Thank you for stopping by.
Running water, so you would imagine a stream or something along those lines. And biofilms are found in damp places. So if you have a pet and you have their water bowl, well, once you take off the water that's not there, you will see like there's a little slimy substance on the bottom of it. That's what you would consider biofilm. They're also found on your teeth and on the bottom of huge ships. So in this experiment, we use kanamycin. Kanamycin is an aminoglycoside which stops protein production in bacteria. And we saw the interaction of kanamycin with E. coli and Serratia marcescens in both these planktonic and biofilm forms.
So clinically, it is commonly known that biofilms are, when they grow, they are way more resistant to antibiotics than they are in a planktonic state. So the purpose of this study was to test the effects of the antibiotic kanamycin on both mixed species cultures in planktonic and in biofilms. So I will go into the planktonic minimum inhibitory concentration of our results, which are in figure 2. So to get into more detail than from the poster, our first tube is just media, which makes it our negative control. And our second tube is media and bacteria with no antibiotic to make our positive control. that's 0. So we set up concentrations from 2, 4, 8, all the way to 1,024 of antibiotics. So it's increasingly in concentration. And in tube 0, all the way to 1024, we added the same amount of bacteria into all of them. So what we wanted to see was the moment the antibiotic would inhibit the bacteria. So the moment it stopped the growth. You can see on our 4th tube, I think it's 4th or 5th tube, of 8 micrograms per milliliter, it shows clear. So the antibiotic inhibited the growth and it stopped it. That is our MIC.
So the next we do is conduct what's called an MBC, or minimum bactericidal concentration. This is when the antibiotics kill the bacteria rather than just inhibit its growth. So when we took samples from each of the growth tubes, so from 8 all the way to 1024, we took samples from that, we plated them, and we saw that slowly as the concentration of the antibiotic was going higher, less and less bacteria was being seen in the plates. And there was no growth in this one, which is 512 micrograms per milliliter. So that would be our minimum bactericidal concentration. So when we did this, if you can tell in this plate right here, we didn't get what we needed with Serratia and E. coli. Serratia usually grows at room temperature. It has a red pigment, which you can see here, or in figure 1 right here. So Serratia didn't have that pigment, so we had to do other biochemical tests to figure out which organism this was. That's figure 4 and figure 4 right here. Figure 4A shows controls. So the first plate here, which the cursor is at right now, is E. coli. When this plate is called EMB plate, and when you plate E. coli on it, it grows with a green sheen. Other organisms that aren't E. coli, they tend to grow either black, or they have a little bit of pigment on them, but E. coli is the only one that shows the sheen there. If you look at our figure 4B, you can see that at high concentration, this is 512, we only see Serratia. And when we conducted it at lower concentrations, we also saw only Serratia. So figure 5 here shows our biofilms data. It shows for biofilms, we test the integrity of the biofilm. To do this, we do a colorimetric assay called the colorimetric assay, which uses the dye crystal violet. You add it into a plate, and you put the plate with your cells into something called a photospectrometer or a plate reader. The photospectrometer then shines a light through the sample, and since the dye is there, it absorbs a certain amount of light, and the light that passes through is what we call an absorbance rate, and that's what you can see is graphed here. This is optical density absorbance at different concentrations. So in biofilms, we saw that at lower concentration, like below 4 micrograms per milliliter, we still see a little bit of growth in the antibiotic, or sorry, in the bacterial growth. But at 4 micrograms per milliliter here, it drops significantly, and the integrity of the biofilms being disturbed. And after that concentration, the biofilms come back, they have more integrity, and they start to stabilize, which can be seen here. This just told us if the biofilms are present and are working in good order, but we didn't know what was going on inside the biofilms themselves. To do that, we plated the samples and saw the result of growth, what happened. That can be seen in figure 6. In figure 6, we see that serratia is more commonly being found. Sabrina, you want to elaborate on that for me?
Yeah, absolutely. So what the point of plating was to see if bacteria tend to either help each other or they tend to hurt each other. So we wanted to visually put that. So what we did was put them on plates. In the lower concentrations, you see more red going on. So unlike the pyctonic, the pigmentation of serratia was not affected here. So we could clearly see that serratia is more dominant in the lower concentration simply by there being more of it. So that was the control plate, which is 0, and then 8. The last plate to the far right is higher concentrations of all the way to 128 micrograms per milliliter of the antibiotic. You don't see as much red here as in the first two plates. What you see here is more white. And again, white colonies represented E. coli here. So overall, they seem to be I think helping each other. Yeah, they seem to be helping each other's growth because, I mean, the bacteria still survives. The bacteria is still growing through the concentrations.
So in conclusion, we know that bacteria is being, we know that in single species that planktonic cultures die off really quickly. And in biofilms, they are still surviving even at those species. We have also considered future directions for this experiment where we might think of adding a different stressor, such as another antibiotic, or what is known as bacteriophages, which are bacterial viruses, which would affect the interactions between them bacteria differently. And we've also considered adding another bacteria in general, which would also change the interaction between them. Thank you, guys, for listening. Do you guys have any questions?
Yes.
I do have one. It says like your purpose is to assess, the E. coli and all that, but what does this, like knowing this, the information that how would you apply it to like now to, do you understand my question? I don't know how to word it correctly.
So how does this apply to, let's say, a clinical setting, right?
Yes.
Okay, so right now we see that a lot of infections, so these lot of infections are multi-species. So we see that more and more infections people are getting are multi-species infections. So knowing that these concentrations of that it requires this amount of concentration of antibiotic to deal with them is necessary, because antibiotics, while they don't hurt human cells, like they don't target human cells, they are toxic to the human body, because most of the human body is made of bacteria. I think it's 90% of the human body is bacteria, while the 10% is human cells. So it does affect our gastrointestinal system. It affects other organ systems. So knowing this is important. We need to figure out how else we can deal with these bacteria's infection rates and other things in that nature. We've also seen that, and this happens with transplant, transplant parts, I guess. So
Right.
heart valves and stuff, biofilms form on those. So
Can you explain what a biofilm is?
So a biofilm is a community of bacteria. So it forms on like damp surfaces, such as if you have a pet, you would have a watering bowl for the pet, right? And when you remove the standing water from the bowl, you see like that slimy substance on the bottom. That's what a biofilm is. And it's just a community of bacteria. They're communicating with each other about what, if there's food there, if there's what surviving condition there are. They're all in constant communication, and they do this by something called quorum sensing. So they release proteins out, and if they get a response back, and they might release a certain protein to get food or share food, share nutrients, and all of that stuff. It's actually pretty cool. We recently found an article that compares biofilms and beehives. I think Sabrina can talk more about that. Right after I finish my thought, I'm really sorry.
No.
So these biofilms form on replacement heart valves and other replacement parts like titanium knees and hips, right? So if you put that into, let's say, a titanium knee or a titanium hip is going to an older patient, if you put that in an older patient, and if it's an infectious bacteria that is pathogenic, it will wreak havoc through their systems.
Wow.
So from what I've researched previously, read articles about in this research, is they tend to just keep these materials in either completely wrong. They do to sterilize them. They put them in high concentrations of antibiotics. Well, as you can see from our data in the graph, the figure 5, right? High concentrations of antibiotics, the bacteria start getting used to it. So the biofilm just starts reforming, and that makes that leads to antibiotic resistances in other situations as well.
That's interesting. I didn't know that.
Okay, Sabrina, you can go ahead and explain the biofilms and
Yeah, so it was an analogy made. Was trying to look for my presentation on it, but I couldn't find it on time. So the way bacteria communicate with each other in biofilms was compared to an analogy to beehives and bees communicating. I honestly cannot remember. Do you remember anything? If you give me a few minutes, I will pull it up.
No, you're good. I mean, I know a little bit about beehives. I like I don't like bees, but I like bees. I like learning about them. So I know that the way they communicate is through their buzzing with their wings flapping. So the way that you just brought it up made me assume that it's obviously they're not talking because they can't vocally communicate, but they're in ways just like the movements, they understand it. So I kind of get it. Like I don't know much about science. Like I love science as a kid, but I don't know how to understand. Like I don't know. Like I was trying to read the poster. I was like, okay, but you explaining it actually helped a lot.
So from what I remember, it's like the bees require a certain amount of quorum or something for them to react to a situation. And biofilms react the same way, where a whole lot of bacteria needs to agree before they actually act on it.
Mm. How did you guys collect the E. coli?
So we just have to order it.
Really?
Yeah.
Is that
So
easy?
yeah. So this E. coli strain is, I think, E. coli EK E2 or sorry, E. coli K12, and that's like one of the oldest E. coli strains that people have just isolated from someone's stool, and that
Because I
just
know there's
continue
been
on.
outbreaks of E. coli lately in
Oh,
lettuce and stuff like that. So I was wondering if you guys got it from like a lettuce or something.
no. So we don't work with pathogenic strains. So if we ingest this E. coli or accidentally, we won't get affected by it.
Interesting.
Yeah. So usually you just the lab technicians for that are there for us, Mr. Frank and Mr. Esmilian. They tend to provide us with E. coli if we need it, and the school just orders these strains of bacteria that we can use.
Yeah, pretty much then it's up to us to keep it alive. So every week we treat on a fresh plate. It's fresh fruit, a fresh environment, and they keep on growing.
Wow.
Yeah, we cause weekly genocides if you think about it.
Well, this is a very interesting presentation. Thank you so much for educating me because I really didn't know.
Oh, no problem at all. Thank you so
You
much.
presented the criminal justice one, right?
Yes.
That was really interesting too. I was
Thank you.
recently talking to someone about how people from who served their sentence still have the stigmata of being criminals, while the whole point is that they have served
Serve
their
their
time.
time.
There's no need,
Yeah.
like that stigma should be disappeared from it.
Yeah, no, I mean, I present, like, my poster session is at 2:05. So if you want to definitely join, you can, and we could definitely talk about this because I love conversing. But, I don't want to take up your time because you're recording this. So you guys have a good day.
Thank you.
See you later if you decide to join my poster. Thank you so much.
Thank you.
Does anyone else have a quote? Okay, he just left. Do you have a question, Karina?
No, I actually, like I put in the chat, you guys did a really good job and I really enjoyed it. And yeah, thank you for the presentation.
No problem.
Thank you.
When you take the standing water out and there's like the slimy substance on the bottom of the water bowl, that's what a biofilm would be. Our experiment is we check the effects of the antibiotic kanamycin, which is an aminoglycoside, so it blocks protein production, on mixed species cultures of E. coli and Serratia marcescens in both planktonic and biofilm forms.
So clinically, it is commonly known that planktonic are less resistant to these antibiotics, but biofilms are way more resistant to these antibiotics. So this could be due to several factors, and we are seeing more and more infections in mixed species. So that's why you see 2 species here instead of 1. So we first performed on the planktonic state. Again, these are free-floating bacteria. These are not attached to anything. Our first tube to the very left is our negative control. So this is only media. Our second control is our positive control with bacteria and 0 antibiotics. So this is our 0 concentration. This is moving on, we did to 2, 4, 8, all the way to 1,024 of antibiotic concentration. So it's increasing in more antibiotics. And in all the tubes from 0 through 1,024, we put the same amount of bacteria of E. coli and Serratia into all of them. So to find the minimum inhibitory concentration, what we call our MIC, we have to find the concentration at which it stops the growth of bacteria. So clearly, our first tube that stops the growth is 8, because 0 to 1, 4 have growth, and then you can see it's cloudy, and the first clip 2 is 8. That is our minimum inhibitory concentration. Now, just because it's clear doesn't mean the bacteria is dead. That is our minimum bactericidal concentration that we have to find. So what we do next is plate all the clear tubes to see where there is no growth of bacteria, and that means that it took that much antibiotic to kill it. So on the bottom figure, on figure 3, we plated low to high concentration. So you can see there's less and less bacteria growing. It proves the antibiotic is working, it's killing the bacteria. Our first clear plate is 512 micrograms per milliliter. That is the concentration it took to kill it. So that is our MBC. Now, the other thing was figuring out the interactions, what was going on in the planktonic state. So what bacteria was surviving in low concentrations, what bacteria was surviving in high concentrations. To see this, we did biochemical tests. I'll further explain this, but with the EMB plates. So on the figure 4, you can see 2 plates next to each other that are the control plates. So E. coli grows a green sheen on this EMB plates. You can see the green sheen on the first one. Serratia doesn't do this. So it's a clear distinguishment between these 2. So as we plated the colonies, small and large colonies on these plates, we didn't see any green sheen in any of these. It showed that Serratia was not only dominant, seems to be the only one growing or taking over E. coli.
Okay. So what are the diseases associated with these bacteria?
Uh, so, okay. With E. coli, there's a lot of uh, gastrointestinal problems that show up. It usually causes what's it called? Diarrhea and other stomach aches, and that's the most of it. Like most common strain, the most common in pathogenic strains, they all have those infections. With Serratia, while it is seen to be pathogenic, it's mostly found in dirt. So there's not a lot of infections that we can see with it. We use these bacteria because they're easy to grow. So it's a good model for us to test these interactions.
Oh, okay. Okay, thank you.
So
Absolutely.
going on from what Sabrina said, we did also do biofilm testings. So to test for biofilms, we have to test if at first we have to test if the biofilms are keeping their integrity with the and the increase of antibiotic concentration, and that can be seen by the graph in Figure 5. So to test this integrity of the biofilms, we do a colorimetric assay, which is a absorbance value assay where you add crystal violet to your samples and run it into a machine that's called a photospectrometer or a plate reader. And as the light is shined through the samples, the sample would absorb some of it, and that is the value that we're given. And we can, looking at that value, we can tell what's going on. So if the concentration of crystal violet is high, the absorbance ratio will be high. So if you look at this graph from 0 to 2 micrograms per milliliter right there, you can see that the antibiotic is the bacteria are still growing. The integrity is good there. But as we go lower, as we go higher in the concentration, around 4 micrograms per milliliter, we see that the integrity of the biofilm is decreasing. Then then it increases back up again and goes back down at around 16 micrograms per milliliter. And then the integrity of the biofilms then stabilizes. This shows us that there is something going on within the biofilms since the integrity is changing so much, but we don't know what's going on. To figure out if there's any changes with the cellular composition within the biofilm, we plated the samples. So we plated 0, 8, and 128. You can tell in 0 and 8 that Serratia, which can be distinguished by its red coloring, is more dominant at the lower concentrations. And if you look at 128, at the edge right there, you see more white colonies, which is an indication for E. coli, which is more dominant in the higher concentrations. So you can see that it's slowly changing the effects, or the antibiotic does have an effect on it, even if it's not completely dealing with the biofilm itself. So after this, we also have like future directions that we do want to take this experiment. That we can take this experiment, we can add different stressors, such as a different antibiotic, maybe 2 antibiotics, or a biofilm, or sorry, not a biofilm, a biofilm, bacteriophage, which is a bacterial virus that only affects bacteria. We can also add other bacteria, since both E. coli and Serratia are gram-negative. Maybe adding gram-positive would have a different effect, or and we would be able to see an interaction between all these different kinds of stressors and variables.
Okay. Thank you guys so much.
No problem. If you have any more questions, please feel free to ask.
No, that's it. Thank you.
Thank you.
Have a good one, guys.
You too. Bye.
Thank you for stopping by.
Bye.
You don't have to
So we are available to go over a presentation, or maybe we just want to go over a part of the presentation or poster. Or if you just have any questions, we could answer them.
You guys could. You guys do like a mini presentation over the poster again?
Yeah, sure, no big deal. So our poster is antibiotic sensitivities of Serratia marcescens and E. coli in 2 growth modes. Bacteria tend to grow in either planktonic and biofilm states. These states are planktonics are usually free-floating bacteria that you would find in running water, whereas biofilms are communities of bacteria that are found on like damp surfaces. So if you have a pet, and it has a water bowl, you would see the biofilms. If you remove the standing water from the bowl, the slimy substance on the bottom of it, that's what you would call like that's what a biofilm is. They're also seen on like tooths and like bottom of ships. So 2 major differences between a biofilm and planktonic mode is they have different genetic expression, and biofilms also produce extra polymeric substance, which is just referred to as EPS, which surrounds them and protects them from environmental stress. In this project, we looked at the antibiotic kanamycin, which is an aminoglycoside, so it blocks protein synthesis. Its effects on E. coli and Serratia in single species and multi-species biofilm and planktonic cultures.
So clinically, it is commonly known that in the planktonic state, otherwise known as the free-floating state, they're not as resistant to antibiotics as biofilms are because of the EPS. So, and other factors, of course. So it's becoming a problem that we're seeing more and more infections in biofilms due to the mixed species. So that's why here we use 2 bacteria instead of just 1. So I will be talking about the planktonic experiments, which you can see beginning on our figure 2. So our first tube has our negative control, which is just media, no bacteria, no antibiotics. And our second tube is our positive control with media and bacteria with no antibiotics. So this is our concentration 0. After that, we go through higher and higher concentrations, like 2, 4, 8, all the way to 1,024. And in all the tubes of 0 through 1,024, we use the same amount of bacteria in all of them. So the only thing that really changes is the concentration of antibiotic. To find our minimum inhibitory concentration, what we call our MIC, it's to find the amount of concentration it takes to stop the growth of bacteria. To see this is to see the first clear tube, which is our concentration of 8 micrograms per milliliter. So you can see in the second, third, and fourth tube, they have growth, they're cloudy, and then it shows clear. 8 micrograms per milliliter indicates our MIC. Now, just because it stopped the growth of bacteria doesn't always mean it kills it. So we plated all the clear tubes on TSA plates shown in figure 3 to see when the bacteria stopped growing. So our first clear plate was 512. That indicated our MBC when it killed all the bacteria. But you could see that as it went higher and higher concentrations, it was less and less bacteria growing. Not only we needed to see what concentration stopped, we also wanted to see relations between the bacteria and the antibiotic. So we did differential tests to distinguish them. On figure 4, the differentials we used was EMB plates, these dark colored plates, and E. coli grows a green sheen. Serratia does not do this. It grows like a faint red pink pigment. E. coli grows a green sheen. So we streaked colonies from low and high concentrations of our plates from figure 2, or I mean from figure 3 on these plates, and we found no E. coli present. No green sheen, no E. coli, this was only serratia.
So we then moved on from planktonics to biofilm cultures. In biofilms, we can't conduct the MIG test because we don't have the enough bacteria. They're in a special community, so we can't just do the same test. We do what's called a colorimetric assay. Now, a colorimetric assay works by having your sample in a 96 well plate, and you add crystal violet, which is a dye that will attach to the cells that are present in the sample. You then put that plate into a machine called a photospectrometer or a plate reader, and that machine that emits a light through the sample, and only a certain amount of it's able to come out. That is how we get what's called our absorbance value. So if the concentration of crystal violet is high, not a lot of light will pass through. That would mean that there is a biofilm present. So this test is specifically used to test for the integrity of the biofilm. So if you look at figure 5 here, you can see as even in the lower concentration, it starts by the integrity getting a little bit better and then dropping significantly at 4 micrograms per milliliter. It goes back up around 8 micrograms per milliliter and drops again at 16 micrograms per milliliter. And after 16 micrograms per milliliter, it starts to stabilize as the concentration of the antibiotic increases. Now, since we know that the higher concentrations, the antibiotic is losing its effects on the biofilm, we do see that there is somewhat of a change at the lower concentrations. So to see if this change was just a microbial makeup change inside the biofilm, we plated the different samples at different concentrations. If you look at this image, figure 6, you can see we plated 0, 8, and 128. And you can see that at the lower concentrations and the positive control, which is 0, that Serratia is the more dominant species of bacteria that is found. At 8, you can also see that it's Serratia, and you can distinguish this because of the red colonies. Serratia grows red at room temperature, and you can see that at optimum temperature, and that's can be seen in the first 2 plates. Now, if you look closely at the last plate, which is 128, you can see that there's a lot more E. coli colonies which grow white, which grow white, and you can see there's less of the Serratia colonies. So we can tell that while the antibiotic is not clearing out the biofilms, they are changing the makeup of the biofilms. Right. So from there, we thought of what our future directions for this experiment could be, and we thought that we can change up the variables a bit. So we can change up, we can add a different stressor, such as another antibiotic, maybe 2 antibiotics, or what's called bacteriophages. Bacteriophages are bacterial viruses that only affect bacteria, and we can see if those are changing the interactions between the organisms, if they're changing interactions between just the whole system that we've seen so far. We've also thought of adding different bacteria, so adding another organism, or maybe adding a different kind of organism, and seeing the results from there. Do you guys have any questions? No, maybe, yes. A lot of them.
No, no questions, but it was a very interesting listen though.
Yeah, no problem.
Thank you for the mini presentation.
No problem, man. That's what we're here for. Does anyone else have any questions? Do you guys want me to redo a little bit of the beginning of it? I know you guys missed that. Yasmin, Taj, Khalil.
Uh, the first time I didn't really see, the last time I watched y'all's thing, I didn't really see the whole of it. I mean the beginning. So I was just wondering if I missed something.
Oh, so we can go over the introduction a little bit more again, or we can go over the whole thing if you want.
Just, the intro is fine. Please.
Okay, so bacteria tend to grow in two forms of growth, which are called planktonic and biofilms. Planktonic bacteria is free-floating bacteria that you would find just flowing, you would find in flowing water. And biofilms are communities of bacteria that you would find in standing water, damp surfaces. So if you guys have pets, if you remove the standing water from their water bowl, the slimy substance on the bottom of the water bowl, that's what you would call a bacteria. Sorry, biofilm. They're also found on your teeth and like bottom of ships. So that's like slimy substance there. So what we did was test the effects of kanamycin, a aminoglycoside. Oh, sorry. They also biofilms and planktonic cultures also differ because they have different gene expressions, and they also have biofilms also have extra polymeric substances, which is like sugary substance that allows them to stick to the walls of the damp surface more easily. What we did was we tested the effects of kanamycin, which is an aminoglycoside, which stops protein production on E. coli and Serratia marcescens in both planktonic and biofilm cultures.
Right, so clinically, it's not as commonly seen to see infections in the planktonic state. They're not as resistant to the antibiotics as biofilms are. So there are more and more infections coming about of mixed species. That's why here we use 2 bacterial strains instead of just 1. So Tosh, I don't remember if you saw the planktonic experiment or the biofilms or both. Okay, I have a question. What are the dark plates on the bottom for clarification? So I think you're referring to the EMB plates, Hafsa. These are different biochemical tests, differential tests. So the reason we use them was because in figure 2, we plated the planktonic clear tubes to see what bacteria was surviving and when it stopped the growth. So we had our results, and our results were 512 was the MBC. We also wanted to see what bacteria were surviving, what bacteria was, maybe both that were there, maybe only one of them was there. So on the bottom left, we see EMB plates that plated our control of Serratia and E. coli. When plated, E. coli shows a green sheen. But Serratia doesn't do this. It just grows a faint pink pigment. It doesn't have this. So what we did was we took colonies from lower and higher concentrations and to see which bacteria were available. And it seems that only Serratia was growing throughout the all the concentration of the plates.
So, Nick, for your question, the most difficult step for this is just the time it takes, and to make sure we finish, do the steps as close to perfectly as we can, because it takes a week to conduct one test. So if you start on Monday, you're good, you do fine on Tuesday, and by the time you reach Wednesdays, a small thing goes wrong, that means you have to stop the experiment for the whole week because you can't continue on. We recently had that happen where one of us forgot to turn on what we call a shaking incubator, and we couldn't conduct the experiment this week due to that.
For me,
Uh,
oh, I'm sorry, you can go ahead.
The other thing that I personally find complicated is just the amount of pipetting we have to do, because you have to be sterile and you still have to like go as fast as you have to go as fast as possible with such specific amounts of liquid that sometimes it gets a lot really stressful. Like that sentence, God. Okay, you can go ahead, Sabrina.
Oh, right. Yeah, so I was actually gonna go based on that it's not as bad for planktonic, but the things we're done with planktonic, we have results, so we moved on to biofilms. Right now, we only have preliminary data, but it's by far, for me, the most complicated, I guess, experiment I've done. There's also tons of pipetting we have to do. We have to do washes in the CV assay trays. So we have to take out our, I guess, loose bacteria, planktonic states, what's floating around, just to leave the remaining biofilms that's sticking to the tray. And we have to take media, release it in there, wash it, take it out, and wash it and take it in by changing tips in each time. And that's just one step of like 4 pipetting steps in the biofilm. So for me, that's the most complicated, but it's not hard. And well, once you get in the groove of it, you just play a song and you'll just finish throughout the day.
So we like to go with heavy metal because it has real screaming sounds, and we just pretend that's our brain screaming to us while we're doing this.
Are there any more questions? We have fun in the lab.
Yes, if you guys haven't taken part in any research lab, I would recommend you guys definitely do that. It's one of the most fun things you will be able to do. What? Anyone else have any questions? Want to go over
Question.
something?
What got you guys interested in this specific research?
So I personally am I am a biology major with a concentration in microbiology. So I just love the microbial world. I like seeing the interactions between them, and just when I heard about Dr. Gulati's experiment, I thought that it is clinically really important, as well as the fact that I got to use these machines that I've never used before. So that was my interest, plus looking at the interactions after biofilms and with planktonics. So a thing we saw when we did planktonic cells, which we weren't expecting, was when we were expecting their results to be similar to single species cultures of planktonic cells. So you can see those results here in table 1, where E. coli, the MIC, which is the minimum inhibition concentration, is 2, and the MBC is 4 for both of them. The MBC is the minimal bactericidal concentration. We were expecting the numbers for the mixed species to be similar to that. And when we conducted this experiment, they turned out to be like as you can see, 8 is a lot higher than just 2 micrograms per milliliters, and pretty much the bacteria don't die till you add 512 micrograms per milliliter. So just looking at these interactions, looking at these results was just interesting in that. And for Sabrina, I said do it.
Yeah, so I attended, I believe it was the same conference last year. So I was looking for research opportunities available, and this seemed like the most interesting one. It deals with bacteria, and it seems like there's a lot of steps. So personally, I like being at a lab. I like doing multiple steps, different steps. Even though these experiments take 5 full days, I don't mind. So that was what interested me in research, just being in the lab. I could talk about interesting findings if you like, or elaborate on a question. Praise, thank you for coming. Keishan, thank you for coming. If you guys have any questions, go ahead, Praise.
Yes.
Hi guys. I'm so proud of you, but I have a question
Thank you.
regarding the minimum, I think something concentration, the MIC. Yeah, because I saw you pointed that out, Yunus, and I see that it's quite interesting. But do you think they have some sort of interaction? I mean, E. coli and SM, do you think they have some sort of interaction that makes them even more difficult to kill when they're, mixed, as opposed to when they're separate or they're individualized?
So bacteria always have some sort of interaction. They can either help each other out or they can just target the others. So since if you look at our if we look at the MBC, we see mostly Serratia. We are predicting that at that concentration where MIC is, we stopped seeing E. coli colonies when we played it. So we're predicting that maybe Serratia is using the E. coli as additional nutrition and surviving based on that. Or maybe there's just such a small amount of quantity of E. coli present that we are just not getting it in a plate and it is surviving. And those are like somewhat of the mutant antibiotic. And maybe those are being maybe Serratia is attacking those, or there are multiple different things that can happen within a tube that we just can't see.
Awesome. And do you think they could be forming some sort of like biofilm or, just something that makes them even more like harder to kill, I guess?
So in planktonics, we make sure they're not able to form biofilms. You do this by you put them in a shaking incubator. So what a shaking incubator is essentially just an incubator which has a plate on the bottom of it that just keeps on spinning until however long you have to run your sample. So our method is we put them in the shaking incubator for 24 hours and it keeps on spinning until, or it spins at 150 RPM.
Thank you. Thank you.
No problem. Anyone else have a question? I know a couple of you joined, so we'll redo the presentation if there are no more questions from the people who heard the good presentation already. Anything? 5, 4, 3, fingers gone, 2, 1. Okay. So bye, Brace. Our presentation was on antibiotic sensitivities of Serratia, marcescens, and E. coli in the two growth modes for bacteria. Bacteria tend to grow in two modes. It's planktonic and biofilms. Planktonic bacteria is free-flowing bacteria that you will see in free-floating bacteria that is seen in floating or running water, whereas biofilms are communities of bacteria that you would see in like on damp surfaces. An example of this will be if you remove your if you have a pet and you remove the standing water from its watering bowl and you see that slimy substance at the bottom of the bowl, that's what a biofilm is. They're usually hard to remove. So in this, the difference between planktonic and biofilm cultures is planktonic cultures and biofilm cultures show different genetic expressions. And biofilms also have something called the extrapolymeric substances, which protect it from the environment and different stressors. These extrapolymeric substances are mostly made of sugars, so they're allowed to stick to the walls, stick to their surface that's needed. In this experiment, we tested kanamycin, which is aminoglycoside, and so it stops the protein production. on E. coli and Serratia marcescens.
So in the clinical field, we don't see many infections with the planktonic state. Again, this is the free floating state. They aren't as resistant to antibiotics as biofilms are because of their EPS structure. So the thing is, we are seeing more and more infections with mixed bacterial species in biofilms. So that's why we decided to do 2 bacterial strains instead of just 1. I will be talking about the planktonic experiment, which is beginning on figure 2. So our first tube here is our negative control with that contains just media. Our second tube is our positive control that contains bacteria and media with no antibiotics. So this is our 0 concentration. From there, we went up to 2, 4, 8, all the way to 1,024 micrograms per milliliter of antibiotic concentration. And then from tube 0 all the way through, we use the same amount of bacteria in all of them. So the only thing that really changed here is just the concentration of antibiotics. The minimum inhibitory concentration is the concentration at which the antibiotic stops the growth of bacteria. So it's pretty clear to see the first, second, third, and fourth tube have growth in it. It's cloudy. So the bacteria was not inhibited here. Where it was inhibited was in the next tube of 8 micrograms per milliliter. Since it's clear, this indicates our minimum inhibitory concentration, our MIC. So just because it's clear and it's our MIC, it doesn't mean the bacteria dies or that the antibiotic kills the bacteria. So what we have to do next is plate all the clear tubes to see at which concentration there's just no more bacteria growing. So in the next figure, figure 3, we plated low and high concentrations. And we did see the antibiotic working. There's less and less colonies forming, less bacteria growing until we get to plate 512, where there is no sign of any bacteria. Because there's no sign, there's nothing growing. That is our minimum bactericidal concentration, what we call our MBC. Not only did we need to find the MIC and the MBC, we also wanted to see the interactions between them. So we did biochemical differential test on EMB plates. These are these dark color plates shown on figure 4A and 4B. So E. coli streaked on these plates have a green sheen. And Serratia does not do this. Serratia grows a faint pink, a faint red. It doesn't grow this green sheen. So it's a perfect differential test for this experiment. On these plates, we plated the colonies from low and high concentrations, and we saw no green sheen on any, meaning that E. coli was not present. What seems to be dominant here is Serratia.
So from after we got our data for planktonic, we started moving on to mixed species biofilm cultures. And since we couldn't do the MIC in the biofilm cultures, we did what's called a colorimetric assay. For this, you use the dye crystal violet. You add it into your sample on a 96 well plate, and you put it in a machine called a photospectrometer or a plate reader. Now, how this machine works is it shines a light through the sample with the dye, and if the sample has more dye, the more light is absorbed within the sample and less light goes through it. And that's how we get our absorbance values. The machine calculates all of that for us. And this test is usually done, or this test was done to test the integrity of the biofilms rather than just checking if the biofilms are dying or something along those lines. So if you look at this graph in figure 5, you can see that from the lower concentrations that there's a small peak going upwards in the lower concentration, but it drops at 4 micrograms per milliliter. So that means the integrity of the biofilm is being weakened there. And after that, as the concentrations increase and you can see that the biofilm integrity is starting to stabilize. We can, knowing that something is happening to the biofilm's integrity, we needed to know what the interaction between the two organisms was. We needed to see if there was some sort of change between the two organisms to figure out which one's more dominant. That you can see in figure 6. So figure 6 is what we plated from the 96 well plates. It shows 0 micrograms per milliliter our control, 8 micrograms per milliliter lower concentration, and 128 micrograms per milliliter, the highest concentration we had at the time. You can see that Serratia is the more likely organism in the positive control and 8 micrograms per milliliter by the distinct red coloration of its colony. Whereas in the 128 micrograms per milliliter, we have more E. coli being shown, which you can tell by the abundance of the white colonies. From here, we can tell that while the antibiotic isn't able to completely clear out the biofilm, it is able to affect the composition within the biofilm itself and change the organism from Serratia to E. coli. So for this, we've also considered a couple future directions. We've thought about adding maybe another antibiotic or having 2 antibiotics at the same time. We've also thought about having bacteriophages, which are bacterial viruses that infect only bacteria and infect only bacteria. We've also thought about adding maybe another organism that would change up the interaction between the 2 organisms that we have present here. Does anyone else have any questions?
Thank you so much, Richmond.
Go, but thank you guys so much. Guys pretty much explained everything, and I like y'all's graphs. It looks like y'all have been busy this semester working on this. So yeah, wish you guys best with y'all's research.
Thank you, Luis.
Thank you.
We're done.